Swayam Prabha. Digital India. Educated India.
Welcome to the organ printing course. So in this lecture, we'll discuss how to design a cornea tissue specific Bioink and how a corneal stromal equivalent can be developed by using 3D bioprinting technique. So this is in this lecture, this is mostly our research work done from our laboratory in collaboration with LB Prasad Eye Hospital and also Center for Cellular and Molecular Biology scientists from there. So this is a collaborative work done by our group. At first, let us understand what is the need for corneal tissue graft. What is the need for corneal tissue graft and how we can do that. Like first, let us understand the where is cornea present, what is the structure, and what are the different structures present within what are the different layers of cornea tissue and how it can be. Then we can discuss how a Bioink can be developed and a bioprinting technique can be strategized. First, cornea is the avascular transparent tissue present in the outermost, that is the outermost layer of our eye. So here, what is that is the this is the that black color portion, the circle that you can see. In some case, in some people, it is brown. It can be brown or even it can be blue. But this tissue is mostly transparent, but the color is because of the structure behind the cornea. Cornea is a transparent tissue, and it's also avascular. There is no blood vessels present within the cornea. So the color of the cornea is mostly because of the tissue present behind, that is the iris and other structures present behind the cornea. And so this is in human, the cornea is almost like 11.5 to 12 in diameter, and also it's a dome shaped structure where the the height is steep at the middle and at the center, and the peripheral the height is less. So here it's a transparent tissue, avascular. It acts as a barrier for the other tissues present behind the cornea. So it's a kind of barrier membrane, and it has very high refractive surface because it refract the light back to the retina so that we can able to see because of that thing. And it is, as I said, it is steeper centrally and other than the peripherals, the surrounding areas. And also the anterior cornea is rigid, so because of that it maintains the curvature. So because the cornea it has a typical dome shaped structure, as it can be seen here, steep centrally in the other peripheral area. So here this has a means is the rigidity. Because of the rigidity, it has highly, so that because it is mostly the collagen fibers they present, so they make a rigid corneal surface. So because of that, the cornea can maintain its shape. And this is the cross section of the whole eye, where this is the part of the cornea where you can see clearly this is the cornea, this part. And if you again see cornea under histological, if you do the histology of the cornea, that is a cross section of the cornea, then you can see cornea is made up of consists of 5 different layers. So the top layer is, or the outermost layer is the epithelium, that we can see, that is the present outermost layer of the cornea, where epithelial cells are mostly present here. So, and this epithelial cells, they present tightly so that a tight layer of epithelial tissue is present. So this layer is almost close to 50 or 30 to 50 micron in width. And then the next is the acellular layer present between epithelium and the stroma. Stroma is the major part of the cornea. Almost more than 75% to 80% of the cornea is made up of stroma. So, but in between stroma and epithelium, the tissue is the Bowman's membrane, where the Bowman's, which is an acellular layer, is mostly basement membrane type of matrix components are present, where it separates the epithelium and the stroma, this Bowman's membrane. And then the stroma is the major component of the eye, or of the cornea, of the cornea. And here the resident cells are keratocytes. These are the keratocytes you can clearly see. So these are the keratocytes that is present, and they maintain this kind of tissue structure of this stroma. Then the innermost layer of the cornea is the endothelium, where endothelial cells are lined up, and these cells are lined up. Endothelial cells has very special function. They maintain the osmotic balance of cornea. They can pump out water if the cornea is highly hydrated. So they maintain the hydration of the cornea. Because of that, also the transparency is maintained the cornea. Now, in between endothelium and the stroma, there is also a acellular layer, that is Descemet's membrane. You clearly see here the Descemet membrane here. So this Descemet membrane is also an acellular layer. No cells are present here. It's also made up of some Descemet membrane components like materials, and it also separates the endothelium and the stroma, so that the stromal cells and there can be a separation present here. So this is the overall the histological characterization of the stroma, or the histological characterization of the whole cornea, including stroma, epithelium, and the endothelium. Now let us see why the cornea is transparent. If you see composition wise, cornea has also made up of collagen and other extracellular matrix component, but skin is also made up of collagen, but skin is not transparent, but cornea is transparent. So because of the arrangement of the extracellular matrix component, and also the particular the order that is maintained by this extracellular matrix component, and also the some due to the physical, the due to the composition, the chemical composition of the cornea is such that it will made, because of that the cornea is transparent. These are the factors, anatomy factors, that helps in transparency of the cornea. Like first thing is the tear film that is present outside the cornea, where we see all the time a tear film is maintained or present over the cornea all the time. So tear film also maintain the help in getting the transparency. Then so because this tear film is as as a it is also between the cornea and the ear, so it has as a film. So this film protects the cornea. The next is the epithelium. Homogeneity is very important. The epithelial cells maintain a particular order. Because of that, a homogeneous arrangement of epithelial cells is very much important on the corneal surface. So because of that, the cornea is transparent. Then the corneal stroma, where the collagen is present in orthogonal. The orthogonal arrangement of collagen is very important, because if you see in one layer the collagen, these fibers are parallelly present. The next layer the collagen fibers, again they are parallelly, but that the orientation is almost 90 degree to the preceding layer. So this kind of orthogonal arrangement of the collagen fibrils is very much important for corneal transparency. The other thing is there is no vascular tissue. So no vascular network present within the cornea. That also helps in corneal transparency. If in some cases, if the cornea become vascularized, then also the people get blind because of this vascularized cornea tissue. Then the other physiological factor that helps in the transparency of the cornea is the hydration. Because the cornea tissue is such that the endothelium, they maintain the osmotic balance, as I said, or the hydration of the cornea. So because of that, also corneal transparency maintained. Now the collagen fibers, there is this some specifications of the cornea is collagen fibers. They have a typical regular lattice-like structures. So because of that, the transparency is maintained, and they are separated by a less than wavelength of light, like 400 to 709 nanometer of light can easily pass through the cornea because of this kind of arrangement of collagen fibers. And also they destroy the scattered light, so that the focused light can only go and reach to the retina, so that we can see the structure. Now, cornea tissue, most of the cases, cornea is fine, but there are certain instances like due to trauma, like injury or burn. It can be chemical burn or it can be of other burn, and also some due to some physical injuries. Some certain cases, also certain cases, some bacteria or viral infection or fungal infection can lead to corneal blindness. So corneal blindness is the fourth leading cause of global blindness. You see, and in India, almost like around 7 million people who have poor vision in at least one eye due to any one of these corneal diseases like either keratoconus, wound scar, infection, chemical burn, inflammation. Due to these different diseases, the cornea can become opaque or the vision can be lost. And now the treatment for many of these diseases is transplantation. And if it is transplanted, then definitely it has to be taken from somebody, like so the it has taken from the corneal donor. And then so that is the gold standard till now, because there is no alternative treatment available for this thing to restore corneal function this by any other means. So there's a clear need to for development of some solution in this for this problem. And if you see the statistics, only 1 in 70 of the needy patient they get mostly they get transplants. So that only 1 in 70 patients. So 69 patients out of 70 they just left untreated, or they just left without any transplant. So around this is the number 12, around 12.7 million people are waiting just for corneal transplantation. There is a huge need or huge demand of corneal transplant, but the supply is very less. So typically there is a requirement of a fully functional corneal tissue with fair amount of accuracy has to be developed to bridge the gap between the demand and supply. So there are various materials people have tried like silk, PLGA, and other gelatin and collagen and other materials have been tried. People have been tried to use for corneal as a to generate corneal graft. But the major challenge for cornea is the transparency, because most of the material they end up getting opaque upon implantation, and so that the blind, so the vision again the vision can be lost. And so that because of the transparency loss in because of the loss in transparency, so that's the major challenge. In our laboratory we work on cornea, we work on digitalized tissue matrix hydrogel. That means when we collect the tissue directly from the, either it can be human discarded or animal tissue, those are not useful, and those are basically not not no, those are not of transplant grade. So we collect those discarded materials, discarded tissues, and then we use a process called decellularization, by which we can throw out all the cells from the material, and we can prepare a acellular matrix by which we can by only the cells can be thrown away, because it is found that the cells are mostly immunogenic in nature, because they have the typical subsurface proteins. Those can act as an immunogen, and they can be immunogenic, and because of that the graft can be rejected because of that thing. So what we found that if we can successfully remove the cells, the extracellular matrix material that can be very well used, and those are not immunogenic, because during evolution these proteins are mostly conserved, and they from one species to another species. So we can very well use that decellularized extracellular matrix materials if we are successful in removing the cells. So in this work, actually what we have done, we have collected the cornea from the eye bank. Those are not of transplant grade, so those are discarded human corneas, and those are mostly used, either it can be discarded or it is used for research. So we have collected those corneas, and then we have used the process for first decellularizing them, then solubilizing them, and then preparing this hydrogel from that discarded cornea. So these are some details from that work, where we have worked on both human cornea and bovine corneas, And then what we have done, we have first we have collected the corneas, we have washed them thoroughly, then we minced them, we have cut them into small pieces, very small pieces, and then we used a process called dissolution, when we treat them with a particular salt solution to, so that the all the cells can be lost. So this after thoroughly after doing this dissolution process, and then we wash them also again carefully, so that the then we can disinfect them, and then we freeze dry them or lyophilize them, so that we can get this suppose this is cornea tissue matrix flakes or powders. So now these flakes we have again this we have use these flakes to solubilize them in acidic solution, and then after pH adjustment we get a hydrogel. So that hydrogel is like a temperature sensitive material, because that hydrogel can flow at less than 15 degrees centigrade, but if we keep it at 37 degrees centigrade, this will become gel. So that so that hydrogel, the hydrogel is a thermosensitive material, so we can use them definitely very well. We can use them for bioprinting application. But before that, we have also characterized this discorized human or bovine tissues. First, by histology, you can clearly see that here the native tissue, where all the structure, all the different characteristic layers are present, and also the cells, you can clearly see the cells present within the stroma. But when you visualize them, this architecture is lost, and mostly what we got some, mostly it is the subfibular proteins and other things are present in the ECM, that those things are there. The cells are gone here from here. Then also we have characterized them by different component like collagen, GAG, and also DNA. DNA quantification is an estimation by which we can understand what is the level of resident cellular content present in that cornea. Like here in the native cornea, if we convert them into 100%, if we consider 100% DNA is present in the native cornea, then almost like less than 3% or 4% DNA is present in the native, in the dislodged cornea tissue, and that is less than 50 nanogram per mg of dry tissue. So it is well below the limit of the DNA that can be used as a transplantable material. So we have successfully dislodged this cornea with our developed process. And then other thing, we have also analyzed two important components of this collagen, important components of the cornea, like matrix component, that is collagen and GAG. Collagen found almost 50% collagen is retained, and GAG, there's a high amount of GAG, almost more than 70% amount of sulfated GAG is present within the cornea. So that's a very good thing, because GAG is the most considered as a reservoir of growth factors, bioactive factors in the resident tissue. So the GAG, if the GAG content is high, typically the bioactive component will be higher in the cornea, in the disulized cornea. So this is for human disulized cornea matrix, and this is for bovine disulized cornea matrix. In both the cases, we found that a good amount of GAG and collagen is retained, and we could successfully remove the DNA, and that is an indication that we can successfully destroy the tissue. Then we have prepared the hydrogel, as I mentioned. We have mixed that lyophilized cornea tissue with lyophilized and disulized cornea tissue in acidic solution, and then again after that we have maintaining the temperature below 15 degrees centigrade. We have optimize the pH or modulate the pH, adjust the pH to 7, and when we adjust the pH to 7, it become a temperature sensitive material, because the collagen, because of the presence of high amount of collagen, it can behave like a self-assembling material, and it can what it can do, and it can also be temperature sensitive material. It can self-assembly, it can become gel. So that's why we need to maintain the temperature at below 15 degrees centigrade during bioprinting thing, so that it should not become gel. And this is the clear viscosity, shear rate viscosity curve, where you can see that the viscosity comes down with increasing shear rate, as I have mentioned in my earlier lectures. Also that this is typically a shear thinning property. Typically it's like a shear thinning material. So shear thinning material is very good for bioprinting. We have already discussed this before. The next thing is the also the gelation kinetics of this thing. As I mentioned, at 37 degrees it will become gel. So if you see at up to 25 degrees, there's nothing happening. Almost like this curve is flat. So that is behaving like a liquid fluidic material, and then at the temperature increasing from here, it becomes gel at 37 degrees centigrade. So the same thing happens for both human and bovine DCM matrix. And also there is a concentration dependent viscosity. That means that for 1% DCM solution, the viscosity is very less, but at 3% the viscosity is higher. It's almost like 10 times higher viscosity can be achieved by increasing the concentration of this Cornea matrix hydrogel. So then the other important thing is the for extrusion based bioprinting, we have discussed this before. We know that for extrusion based bioprinting, shear recovery is the very important thing, because when you force the material to pass through a very fine nozzle, then that time the material is under high shear rate, and then the viscosity generally comes down. But again, when the material is out of the syringe, that's again zero shear viscosity, zero shear rate, and that time the viscosity should again be should become high. And so we should, by doing this shear recovery test, we understand that what is the recovery of this thing. Like at when the shear rate is very high, at that time the viscosity will be very low, and again when the shear rate is low, with the viscosity will be higher. So is that almost like up to 90% recovery can happen by after one cycle. That's what we found here. So both these hydrogel, they show similar behavior in this for shear recovery properties. We have also done some few physical characterization, like the porosity estimation and the swelling and this swelling property, because these are very important properties for the hydrogel and also for the bioprinted corneas. Like the first thing is the porosity. Here we can clearly see that the hydrogel has very high porous structure than the only lifeless tissue, and it's clearly so because this is the lifeless tissue where the water content is very less, but in this case of DCM hydrogel the water content is high. So that's why there is a clear difference between these two in terms of porosity. And other thing is, surprisingly, when the DCM hydrogel is put into PBS or media, we saw some amount of deswelling, rather swelling in the material. This here actually the compaction of the material happens, and the drainage of water that happens, and because of that thing, clearly there is almost like 60%, and somewhere it is getting stabilized to 62 to 3% of deswelling we could see over 8 days of incubation.